everyone, welcome and thank you for joining us today. For those who don't know me, my name is Mabel and this is Mahir. We are from Auckland Bioengineering Institute and we are part of SPARC MAP-Core team. Today we will take off from where MBF left at part 1 of the webinar series and continue to explore the pipeline of scaffold registration using segmentation data. For any of you who missed part 1 of this webinar, or wishes to revisit it, you can find the YouTube link at the end of this presentation. We also have our team here with us today, so please feel free to submit any of your questions throughout the webinar and we will address them at the end of this presentation. To submit questions, simply click the orange arrow button to open the Go to Webinar Control panel. Find the Question To panel. Type your question in the box and click Send. You can also undock the question box from the panel if desired. As a quick recap from Part 1, Auckland Bioengineering Institute and MBF Bioscience are both part of the SPARC MAP-Core team. We work closely with different experimental groups funded under SPARC to create 2D and 3D connectivity maps of the autonomic nervous system. And these maps are made available on a public data sharing platform called SPARC Portal. The SPARC Portal contains maps which are useful for exploring the diverse experimental data generated by SPARC investigators, which are curated and made available for download. The portal also acts as a public platform for storing and updating models with these curated experimental findings. The portal contains maps which under the SPARC MAP-Core pipeline, experimental data such as microscopy image data typically undergoes the image segmentation process using tools developed by MBF Bioscience as discussed in Part 1 of the webinar. The segmentation data then reaches us at Auckland Bioengineering Institute, where our team will register the segmentation data onto organ scaffolds and provide visualization tools on the SPARC portal for users to interact with these data. Apart from image data, we also deal with non-image data such as RNAseq, electrophysiology measurements, motility, and pressure volume data. However, today we will be focusing on how image data is registered on scaffolds, and we will discuss how non-image data is handled in a future webinar. So you might wonder why your data should be registered to a scaffold. Throughout this presentation, it will become apparent how scaffold registration allows visualization of image data on 3D organ scaffolds. Registering data onto a scaffold helps bring data into a common coordinate framework, which enables cross-species or inter-subject comparison, and allows comparison of data both within and between experiments. This is a very powerful tool as we now have the ability to piece data from seemingly unrelated studies together onto a single model. This allows for the interrogation of possible interaction between different experimental findings and also help drive further research by identifying gaps in our current knowledge. Right now, we have scaffolds for the 5 major organs, namely bladder, colon, heart, lung, and stomach. Each of these organ scaffolds is essentially a 3D finite element model which captures the anatomy of the organ. We have developed these scaffolds using our in-house software called ScaffoldMaker, which is built on OpenCMISS libraries. These scaffolds are currently set up as generic models, which represent the average shape and size of each organ. But ScaffoldMaker also provides the capability to parameterize these scaffolds using a relatively small number of anatomical characteristics. And this enables the representation of organs for different species, subject, and samples. We have also recently submitted these generic scaffolds for publication on the SPARC portal, and as their curation is completed, they are becoming available on the portal. So now we will demonstrate how a scaffold can be customized for specific species using the colon scaffold. The colon scaffold is designed based on a central path concept. Basically, our tool allows us to create a path of a colon which can be traced from a photograph, CT, or MRI image. Our tool then uses that line to generate a scaffold which follows the exact same path. We have also created building blocks called segment profiles, which captures the characteristics observed in the cross-sectional profiles of colon from different species. For instance, mouse colon presents a tubular structure due to the absence of Tania coli, whereas both human and pig have a more Haustra-sac appearance due to the presence of Tania coli. We can also control dimensions such as colon diameter, wall thickness, and Tania coli properties, as well as handle variation of these properties throughout the entire length of the colon. So by using all these users' inputs, a scaffold specific to a species can be created by assembling all these building blocks together along the path of the colon. I will now pass on to Mahir, who will discuss how scaffolds can be fitted for a specific individual.
Thanks, Mabel. So as part of the scaffold mapping pipeline, we have developed tools to customize our scaffold to individual datasets. Scaffolds contain distinct regions and landmarks, annotated with standard anatomical terms based on the ontologies provided by the SiteCrunch infrastructure from the FAIR Data Informatics Lab at UCST. We work with the researchers who produce the data and our colleagues at the MBF Bioscience to ensure that scaffolds and data have the required corresponding annotation. This allows for an easy and automated registration of data to scaffolds. So for the purpose of this presentation, I will be using this data set shown here with permission from the SPARC investigators to show you how a rat heart image registered into our scaffolds. Here I have the segmented image data and a corresponding rat heart scaffold, both showing matching annotation groups. Since in this data set we're interested in the atria of the heart, we focus only on these groups and remove the ventricles. So using our software scaffold fitter, we initially align the scaffold and data so that they are in the same spatial location. The fitting process then finds the corresponding anatomical locations of the data on the scaffold and then morphs the scaffold. The result is a scaffold that has been fitted to represent the exact same anatomical points as the data, labeling the same piece of tissue across all configurations for an individual or an equivalent anatomical location across multiple individuals. I should probably mention that the scaffold fitter software allows for parameter tuning in situations where data is distorted and fitting is difficult. As you can see here, there are a number of parameters which regularize the fitting by controlling the, for example, the amount of deformation and or curvature or other things, just to ensure a smooth shape is produced at the end. Now that the fitting is done, ICNs can be easily mapped on the scaffold. ICNs are these cells labeled in yellow and are mainly populated around the atria and pulmonary veins. Once these cells are mapped, we can transform the scaffold, the fitted scaffold, back to its so-called generic form, so that the shape is more representative of its in vivo configuration. Remember, because the surface of the fitted scaffold now represents the anatomy of the data, the coordinates effectively identify the position of any tissue particle independent of its location in 3D space or how distorted it is. So, on deforming the scaffold back to its in vivo configuration, moves any mapped ICN data while respecting their anatomical locations. So at the end, this process will now allow for comparison across different subjects by bringing all the data into one integrated scaffold. And that's the beauty of it all. You now have a framework into which many different aspects of tissue structure can be assembled. For example, the spatial distributions of ICN data as shown here. But it's not limited to that. You can have other structures such as muscle fiber orientations, vascular geometry, or neural pathways. So just to summarize, our job is essentially to work with different people from different organizations to register experimental data into our computational scaffolds, curate the results, and then publish them onto the SPARC portal for everyone to access.
So some of the image data that we work with are obtained from samples abstracted from flat preparations of organs. For example, here we have a vascular image from a mouse colon section, which was obtained by Dr. Martha Howard. To map this image data to a 3D scaffold, we first segment the image to obtain the coordinates and the size of the blood vessels. The image data is then processed to straighten out the uneven cut edges. And this step is essential as we need to ensure that the sample can be aligned nicely along the straight edges of the flat colon scaffold. The data is then mapped onto the correct location on a flat colon scaffold using the information of where the sample was collected from. From here, the scaffold and its embedded image data can be folded back into its 3D configuration. This allows any image data, be it a neuron network or a vascular image, to be mapped onto a common coordinate framework for visualization in a 3D environment. Data from different experiments can now be brought together onto a common platform, and investigators now have the opportunity to explore how different data sets can interact with one another. And this helps foster potential collaborations to further our current research of the autonomic nervous system. Even for data derived from the same experiment, such as the one shown here from Dr. Tashi's lab, registration of their data onto a pig colon scaffold has helped put the data set into perspective. In this example, we have mapped the image data of the inner submucosal plexus along with the transmural section, and we anticipate that as we continue to map samples obtained from other transmural layers, we are looking at a thrilling possibility to study how the pattern of neuron networks evolves transmurally, and hopefully gain a closer insight as to how neurons can interact across the different layers. This highlights the beauty of scaffold registration as we now have access to a platform for investigators to continually improve and update models of nerve organ interactions as discrete pieces of data continue to come together on a common framework. Although we have used the heart and colon as our main examples to illustrate our scaffold registration process today, please note that this process is not limited to these two organs. We are also currently applying similar techniques to map data onto our stomach, lung, and bladder scaffolds. We hope that you now have a much clearer picture of the steps involved in scaffold registration, and you are as excited as we are about the benefits and potential that scaffold registration can bring. We certainly look forward to registering more of your data onto the different scaffolds. So please feel free to get in touch with us through the MAP-Core support channel on Slack if you would like to have a chat about what we can do with your data. We would also like to take this opportunity to thank our collaborators again for allowing us to use their data for demonstration today, and thank you all for your attention.
And so now feel free to send your questions in the chat area here if you have any.
Yeah, hello everyone. I'm Li Wang. Thank you for the presentation and very nice images and the settings. I just wonder, all the wonderful images, if the readers want to know what the image was showing, I mean, the structure, for example, the two color-coded group of cells, what kind of cells are they? So it's only looking at an image without labeling the structures of cells. It's hard for the people not working on the subject to know what the contents are.
I see. Yeah, thanks. Thanks for the question. So really, with the annotation groups and finding the distinct regions is kind of a collaborative work. So we work with the investigators themselves, plus the SPARC anatomy working groups, in order to ensure we find or we can label, annotate those distinct anatomies and regions. So it's like a back and forth sort of process to kind of come up with these anatomical locations. And yeah, it can be a difficult process for some regions that maybe are not well defined or something like that. But at the end of the day, for at least for the purpose of the scaffold fitting and registrations, we will need some corresponding regions between data and scaffold in order to perform the process. So I mean, at the end, ideally, we want to have the correct anatomies for both data and scaffolds. But sometimes, for example, if it proves difficult or impossible to identify a specific region, then we can have pseudo anatomical landmarks or just some user kind of a specified annotations so that we can actually perform the registration. Does that make sense?
Yes, thank you. I mean, may not be necessary to label every details. I mean, at least the labeled cells, neurons, again, the two color-coded groups of cells in the heart, at least mark those, what kind of cells they are. Most people know it's in an angiome, so may not be mark everything, especially the common knowledge, at least for the special labeling.
Sorry, so if I get your question correctly, so so the question is whether whether it's possible to label individual sorry, I'm just writing out visible on the scaffold on the portal.
So I think what's happening is in this demonstration, we did not show how we annotated and how we're going to present these annotation groups. So as long as you are able to annotate the cell groups that you want to see when we are doing the segmentation, we will be able to import this onto when we do our scaffold fitting process. And then we can present it on the portal as well. So when it show up on the portal, when somebody click on a specific group of cells, the name of that cell will show up in the legend as well. So it will be color coded and the corresponding name will be on the platform. And people will also be able to search based on the name of the cell group. Does that make sense?
Yes, thank you.
There's another question. It says, was not clear from the demonstration how tissue layers and compartments are represented in the scaffolds. Could you comment? Maybe did you wanna?
At this stage, we haven't at least for colon, for colon scaffold, we haven't marked out the different tissue layers, but if we have like information about the anatomical location of how these, like what is the spacing between the tissue layers, or like special landmarks that we can use, we can actually quite easily mark it out in our scaffolds.
Yeah, for example, we didn't show in this presentation itself, but for example, bladder and stomach, they because we have information about the individual layers. For example, in bladder, we can have the information from the luminal layer and the serosal layer, and the stomach has multiple layers from mucosal to all the way to serosal and all the muscular layers. The scaffold actually allows for creating these layers if the information is available or is given by the image. And so that's one of the features of scaffold. And also regarding the previous question, Richard Christie, the developer of ScaffoldMaker, also commented in the chat. It says, we're still developing the visualizations on sparc.science, and we're open to user needs here. There's much scope for controlling integrated visualizations and labeling of parts. Thanks, Richard. So I hope that also clears. Now, do you have any other questions? In regards to the heart, is there a scaffold comparing the ICN between rat and human readily available? So at this stage, we don't have that data available. The currently available on SPARC portal is the data set I showed here, which looks at different specimens from rat species. And I mean, in future, as data becomes more available, so we can obviously provide that kind of information as well, depending on the research needs. And yeah, but I mean, in terms of possibility, it's completely possible, but it's just not on the portal at this stage. Thanks everyone again. Please feel free to Slack us or email us if you have any questions. And we look forward to seeing you guys in future webinars. Thank you.